Hello everyone, this is Pratima and welcome back to Planet Physiology. Today we shall discuss MCQs on renal physiology. Most of the questions are PYQs from MGR University and few are from other sources. So let's begin with the introduction to renal system. Which of the following statements is wrong about macula densa? A. These are modified epithelial cells of DCT. B. Macula densa detect changes in NaCl concentration in the filtrate. C. They secrete renin. And D. They play important role in tubulo-glomerular feedback. Macula densa are modified epithelial cells of initial part of the DCT which passes between afferent and efferent arterioles. They act as sensory receptors which detect changes in sodium chloride concentration in the filtrate entering DCT. Decrease in renal blood flow or GFR decreases NaCl concentration in the filtrate and stimulate them. Macula densa in turn sends signals to JG cells to release renin, leading to activation of renin angiotensin system to regulate renal blood flow and GFR. This feedback regulation is known as tubulo-glomerular feedback. Thus, the correct answer for this question is option C, because renin is secreted by JG cells and not by macula densa. Next question. Renin converts A. Angiotensinogen to angiotensin 1, B. Angiotensin 1 to angiotensin 2, C. Angiotensin 2 to angiotensin 3, and D. Angiotensin 3 to angiotensin 4. Correct answer is option A. Renin converts angiotensinogen to angiotensin 1. Conversion of angiotensin 1 to angiotensin 2 occurs by the enzyme angiotensin converting enzyme, that is ACE, which is predominantly found in pulmonary capillaries. Another question on the same topic. Angiotensin 2 causes all of the following except A. Stimulation of thirst, B. Aldosterone secretion, C. Increased ADH secretion, and D. Vasodilatation. So as you know all, angiotensin 2 is potent vasoconstrictor which raises blood pressure, and hence the correct answer is option D. Apart from vasoconstriction, angiotensin 2 also causes secretion of aldosterone by adrenal cortex. It stimulates thirst center and also releases ADH from posterior pituitary. All these mechanisms help to correct hypovolemia and hence the blood pressure. Other actions of angiotensin 2 include increase in secretion of ACTH and decrease in baroreceptor sensitivity. Next question. Normally, albumin is not filtered because A. Its diameter is greater than that of fenestrations of glomerular capillaries, B. Negative charge on the filtration membrane repels albumin, C. Slit pores do not permit albumin to pass through, and D. All of the above. Now, glomerular membrane is highly porous. It allows neutral substances up to 8 nanometer diameter to pass through. Diameter of albumin is 6 nanometer, which should easily cross the membrane. But negatively charged sialoproteins on the glomerular capillaries repel negatively charged albumin. Hence, despite its smaller size, albumin cannot be filtered, and hence the correct answer is option B. In case of acute glomerulonephritis, negative charge on the filtration membrane is lost, leading to albuminuria. Next question on GFR. In case of renal stones, GFR decreases due to options are A. Decrease in glomerular capillary hydrostatic pressure, B. Increase in oncotic pressure, C. Increase in Bowman's capsule hydrostatic pressure, and D. Decrease in Bowman's capsule osmotic pressure. So all these are the various forces that determine GFR. Due to tough renal capsule, normal Bowman's capsule hydrostatic pressure is positive and opposes filtration. In case of renal stones, this pressure further increases and opposes the filtration, leading to decrease in GFR. So correct answer is C, increase in Bowman's capsule hydrostatic pressure. Next question. Nephrotic syndrome is due to A. Increased glomerular permeability leading to loss of proteins. B. Decreased glomerular filtration rate. C. Increased loss of sodium chloride. And D. Increased production of plasma proteins. In nephrotic syndrome, large quantities of proteins are filtered and lost in urine because of increased glomerular permeability. Hypoproteinemia results into edema and decrease in plasma volume. Due to this hypovolemia, renin angiotensin aldosterone system gets activated, leading to salt retention as an attempt to maintain blood volume. Hence, the correct answer is option A, increased glomerular permeability leading to protein loss. Next question. About 90% of filtered glucose is reabsorbed in early part of proximal tubules by A. SGLT1, B. SGLT2, C. GLUT3 and D. GLUT1. Glucose absorption in renal tubules occurs via SGLT2 transporters at luminal surface, that is at the apical border. It is then transported in ECF by GLUT2 transporters by facilitated diffusion, and normally almost 100% of the glucose is reabsorbed by the PCT, as you can see in this table. This table is from Guyton's textbook of physiology. So correct answer over here is SGLT2. Another question on the same topic. Tubular transport maximum for glucose in a healthy young man is about A. 100 mg per minute, B. 225 mg per minute, C. 375 mg per minute and D. 500 mg per minute. Correct answer is option C, 375 mg per minute. In females, TMG value is 300 mg per minute. Transport maximum is the maximum rate at which a particular substance can be actively transported by the renal tubules. It is due to saturation of carrier proteins by the solutes. Beyond this value, the substance is excreted and appears in urine. Next question. Transport maximum is not exhibited by A. Glucose, B. Lactate, C. Creatinine and D. Sodium. And the answer is option D. Sodium. Sodium transport in proximal tubule obeys mainly gradient time transport principle rather than tubular transport maximum. In distal parts of nephron, sodium transport exhibits transport maximum, but this maximum can be raised by the hormones like aldosterone. Here is a table showing transport maximum values for various substances which are actively reabsorbed or secreted. To know more about this topic, watch my video on transport maximum. Next question. What fraction of filtered water is reabsorbed in the loop of Henle? A. 15%, B. 25%, C. 35% and D. 5%. And the correct answer is A. 15%. This value is as per Ganong's review of medical physiology. Here is a table to summarize the important facts about water reabsorption in various segments of nephron. You can pause the video and go through it. Coming to the next question. Which is the fundamental mechanism generating hypertonicity in renal medullary interstitium? A. Active transport of sodium chloride in thick ascending limb. B. Action of ADH in the collecting ducts. C. Passive recirculation of sodium chloride in the medullary interstitium. And D. Urea permeability of collecting ducts. Correct answer is A. Active transport of sodium chloride in thick ascending limb. This is the first step in countercurrent multiplier mechanism that generates osmotic gradient between medullary interstitium and descending limb of loop of Henle, which induces osmosis. Next question. Identify the correct statement. First statement is the renal medullary blood flow is high. This statement is wrong because renal cortex receives about 93% of the total renal blood flow. Option B is Vasa recta creates medullary hyperosmolarity. This is also wrong statement because vasa recta maintain medullary hyperosmolarity by countercurrent exchanger mechanism. The countercurrent multiplier mechanism is responsible for creating the hyperosmolarity. Option C is medullary blood flow is less than 5% of total renal blood flow. As per the values in Guyton, blood flow in the renal medulla accounts for only 1 to 2% of the total renal blood flow, and hence this is the correct statement. The last option is U-shaped structure of vessels do not minimize loss of solutes. Actually, due to U-shaped structure of vasa recta, solute loss is minimized, and hence this also is the wrong statement. So correct statement is option C. Another question on the same topic. Inability of kidneys to concentrate or dilute the urine is called A. Anuria, B. Uremia, C. Oliguria, and D. Isostenuria. And the answer is D. Isostenuria. Anuria is total cessation of urine output and oliguria is diminished urine output. Uremia refers to accumulation of protein breakdown products in the body fluids. Next question. The obligatory 24-hour urine volume to maintain solute homeostasis in a healthy adult male weighing 65 kg and consuming a balanced 2000 calorie diet is approximately. Options are A 100 ml, B 500 ml, C 1000 ml, and D 1500 ml. Obligatory urine volume is the minimum volume of urine that must be excreted to remove water-soluble metabolic waste per day. It is decided by maximum concentrating ability of the kidney and the amount of solutes to be excreted. A normal healthy person on a balanced diet needs to excrete 600 milliosmoles per day solutes, and the maximum concentrating ability of the human kidney is 1200 milliosmoles per liter. Hence, the obligatory urine volume becomes 500 ml per day. Next question. All of the following factors stimulate potassium secretions by the principal cells except A. Increased extracellular fluid potassium concentration, B. Increased aldosterone, C. Increased hydrogen ion concentration, and D. Increased tubular flow rate. Correct answer over here is option C. Increased hydrogen ion concentration. Usually when ECF concentration of potassium increases, more potassium is filtered and most of it is actively reabsorbed by the proximal tubules. So if there is increased extracellular potassium, more potassium should be excreted, and it mainly occurs by raising aldosterone levels. Aldosterone increases number of ENaCs in the luminal surface and stimulate activity of sodium-potassium pump. Thus it increases sodium reabsorption and potassium secretion. Increase in tubular flow rate, especially in the distal segments, also increases potassium secretion. So the correct answer is C. Increased hydrogen ion concentration. Few points to confirm this answer. First, potassium secretion is function of principal cells while hydrogen secretion is function of intercalated cells. And second, potassium secretion is decreased when hydrogen secretion is increased. This is due to competition for sodium in the tubular fluid. Another question on the same topic. Cells responsible for acid secretion in kidney: A. Type A intercalated cells, B. Principal cells, C. Juxtaglomerular cells, and D. mesangial cells. As studied in previous discussion, intercalated cells are concerned with acid secretion. Type A or alpha intercalated cells are concerned with hydrogen secretion and bicarbonate reabsorption, while type B or beta intercalated cells secrete bicarbonate and reabsorb hydrogen ions. You can easily remember this as type A for acid secretion and B for base secretion. To know more about intercalated cells, watch the mechanism of urine formation part 2 on Planet Physiology. Next question. The major nephron site where titrable acid is formed. A. PCT, B. DCT, C. Loop of Henle and D. Collecting ducts. Titrable acids refer to amount of hydrogen ions excreted in urine that are bound to buffers like phosphate buffers or bicarbonate buffers. In PCT, hydrogen ions which are secreted into the tubular fluid combine with filtered bicarbonate as well as phosphates to form titrable acids, primarily two phosphates. Hence, the correct answer is option A, PCT. Next question, again on acid-base balance. Most important buffer system present in distal convoluted tubule is A. Bicarbonate, B. Phosphate, C. Protein and D. Ammonia. Secreted hydrogen ions react with dibasic phosphates to form monobasic phosphates. This happens to the greatest extent in distal tubules and collecting ducts because phosphates that escape proximal reabsorption gets highly concentrated in the distal segments. The reaction with ammonia occurs in proximal as well as distal tubules, and hence the correct answer over here is option B, phosphate buffer system. Next question. A tonic bladder occurs in destruction of A. Afferent impulses from bladder, B. Efferent impulses from spinal cord, C. Facilitative impulses from the brain and D. Inhibitory impulses from the brain. A tonic bladder is also called as tabatic bladder because tabes dorsalis causes destruction of dorsal roots. Damage to sensory nerves breaks the reflex arc leading to loss of tone in the detrusal muscles and hence it is called as a tonic bladder. So the correct answer is option A, afferent impulses from bladder. The same knowledge is tested in January 23 paper. Destruction of sensory nerve fibers to the bladder leads to A. Atonic bladder, B. Neurogenic bladder, C. Hypertonic bladder and D. Automatic bladder. And you already know the answer. It's A. Atonic bladder. Neurogenic bladder is caused due to partial damage to spinal cord or brainstem which removes inhibitory control over the bladder. As a result, there is continuous stimulation of detrusal muscles leading to hypertonic bladder. Hence, it is also called as spastic bladder or uninhibited neurogenic bladder. Automatic bladder results due to complete destruction of spinal cord above S2. This removes only the higher control over the bladder, but the basic micturition reflex is normal. As a result, bladder automatically empties once it is filled and hence the name. Now coming to the diuretics. Following are loop diuretics except A. Furosemide, B. Ethacranic acid, C. Bumetanide and D. Mannitol. Diuretics are the substances which increase excretion of water in urine, or in other words, diuretics increase urine output. They are classified based on their mechanism and site of action. Loop diuretics inhibit NKCC2 transporters and prevent urine concentration ability of kidneys. So in the given options, except mannitol, all are loop diuretics and hence the answer is D. Mannitol. Mannitol is osmotic diuretic. Next question. All of the following drugs inhibit sodium potassium 2-chloride cotransporter in thick ascending limb of loop of Henle except A. Furosemide, B. Ethacranic acid, C. Acetazolamide and D. Bumetanide. So sodium potassium 2-chloride cotransporter is nothing but NKCC transporter in loop of Henle. So here except acetazolamide, all are loop diuretics. Acetazolamide is carbonic anhydrase inhibitor and it acts at proximal convoluted tubule. Coming to the next question. The diuretic that acts by inhibiting carbonic anhydrase activity exerts its effect in A. Proximal tubule, B. Loop of Henle, C. Distal tubule and D. Collecting ducts. Yes, as we have seen just now, Carbonic anhydrase inhibitor acts on proximal tubules. It decreases hydrogen secretion and thus it prevents sodium reabsorption, leading to excretion of excess of water. Next question. Which of the following potassium sparing diuretic drug is an aldosterone antagonist? A. Mannitol, B. Furosemide, C. Spironolactone and D. Triamterene. As already discussed, mannitol is osmotic diuretic and furosemide is loop diuretic. So these two options are out of question. Both remaining drugs are potassium sparing diuretics. Out of this, spironolactone is aldosterone antagonist. That is, it inhibits action of aldosterone on principal cells. As it inhibits sodium-potassium exchanger, it prevents potassium secretions and hence the hypokalemia. So it is also called as potassium sparing diuretic. Triamterene also acts as potassium sparing diuretic, but it inhibits epithelial sodium channels, that is ENaCs, but it does not prevent action of aldosterone. So it is not aldosterone antagonist. So correct answer is C, spironolactone. Now the questions on renal clearance. How much is the clearance of a substance when its concentration in the plasma is 10 mg per deciliter, its concentration in urine is 100 mg per deciliter and urine flow rate is 2 ml per minute? A. 2 ml per minute, B. 10 ml per minute, C. 20 ml per minute and D. 200 ml per minute. So renal clearance is the volume of plasma which is completely cleared of the substance per unit time. Formula for its calculation is UV by P, where U is concentration of the substance in urine, P is concentration in the plasma and V is urine flow rate. While calculating, pay attention to match the units. Plasma and urine concentrations are expressed in deciliter while urine flow rate is given in ml. So first convert all the values in milliliters. You know the relation 1 deciliter equal to 100 milliliters. So change the unit properly, substitute the values and we get the answer. Clearance is 20 ml per minute. Now the last question of the session. Which substance is used for measuring renal blood flow? A. pH, B. Urea, C. Inulin and D. Creatinine. And the answer is PAH, that is para-aminohippuric acid. PAH is filtered as well as secreted by the tubules and hence it is used to calculate renal plasma flow. Based on this value as well as hematocrit, renal blood flow is then calculated. So details are available in my video on renal circulation. Urea clearance is used to measure efficiency of kidneys to handle metabolic wastes, while substances like inulin, creatinine, mannitol, which are only filtered, are used to measure GFR. So that's all for this session. Most of the important topics in renal physiology are covered in detail in my earlier videos. Links for all are provided in the description section below. If you have any questions, do post them in the comment section below and I will be happy to answer them. Are you new to my channel? Then please subscribe it and press the bell icon to stay updated about the new releases. If you like the video, give it a thumbs up and share it with your friends. Thank you for joining in and see you in the next video.